Hello, Lucía. How are you doing? How is
Hi.
everything over there in Japan?
Hello. Very nice. Thank you. It's going so very well so far, and yeah, very happy to be here and chat with you a little while.
Well, thank you for accepting the offer to take on this interview.
Great.
So first, you're currently carrying out a research stay in Kyoto, where you're working. Why is this center considered a world leader in its field, in the place you're working currently?
So right now in Kyoto, I'm working in Kyoto University, and the research center, is the Center for IPS Cell Research and Application. I'm doing an international stay from my PhD in Spain, which is based in cystic fibrosis as a pathology. And here, what we're doing is models of the pathology, cystic fibrosis in this case, that are more representative of the patient's genetics profile. For this purpose, we're using iPS cells, which are cells derived from patients that are reprogrammed to be pluripotential. iPS cells stands for induced pluripotential stem cells. So what we do is we get cells from a patient, we reprogram them to be pluripotent, and then we derive them to the tissue that we want to study. In this case, as cystic fibrosis has a very strong affection in the pulmonary system, we develop them to an airway epithelium. So why is this center leader in the field? Is because the center was founded by Dr. Shinya Yamanaka, who was Nobel Prize in Medicine in 2012. And he was the one who discovered the reprogramming factors to transform a fully differentiated cell back to a pluripotent cell, an induced pluripotent cell in this case.
Uh-huh.
So yeah, it's
It's fascinating.
Mhm.
This technology. What I'm guessing from what you're saying is that this technology is very useful in what's so-called personalized medicine, right? Which allows you to have special treatments for specific patients or specific rare diseases. I guess I think cystic fibrosis classifies as a rare disease. Yes.
Yes, cystic fibrosis is one of the least rare within the rare diseases, but still considered a rare disease. And yes, IPS helps you model a lot, especially genetic pathologies. Um, the thing with studying a human genetic disease is that it's very complex to understand in vitro what's happening in a physiological context, because normally when you're using cellular models, there you only have one cell type. This does not happen in a physiological state. So iPS cells help you have a multicellular structure in vitro, all derived from the same patients. So they're homogeneous genetically speaking, and helps you a little bit understand what's happening and how cells react. It's still in vitro, so you don't have to depend on a human or an animal, but in a more physiological context, or a
Environment.
more physiological relevant environment. Yeah.
Hmm. Great. You also obtain a GSPS fellowship, if I am not mistaken, which is
Mhm.
highly competitive and rarely awarded to Western researchers, as far as I know. What this opportunity mean to you, both professionally and personally?
So, well, just to clarify, within the JSPS is the Japanese Society for Promotion of Science, and it has a lot of grants. The one that I was awarded was a postdoctoral short term grant. which is meant originally for international postdoctoral researchers to come to Japan to do a research stay. It is also exceptionally awarded to PhD students if they're on their last year of PhD and they have an interesting project. And they award 25 grants per call. So it is pretty competitive for all the applicants worldwide. Yeah, so I guess I was one of those non-postdoc exceptions, which was great. And yeah, it's been a great experience so far, both professionally and personal, because of like what means moving outside Spain, going to Japan, being involved in a very different culture, a very different culture inside and outside the research. So that's great. Yeah. And also this grant came with money for research, which is the first time in my life that I have money available for me to do my research. So it has been very nice to have that available. It allowed me to attend a Keystone Symposium in January here in Kyoto, which was the 20-year anniversary of the IPS. And it was a very expensive Congress. So I wouldn't have been able to go any other way, but it was very enriching and very interesting. So it's been wonderful so far.
Awesome. As you mentioned, your work focuses on cystic fibrosis. How does this research connect with your PhD work at Nanbiosis Unit 10? What is the link in these two projects, and what does this stay provide in addition to what you're already doing in the unit?
Okay, so in Nanbiosis Unit 10, what we do is we have two lines of work. One of them is the synthesis, the optimization and production and characterization of non-viral vectors for gene delivery and other types of delivery to cells. And the second branch would be 3D bioprinting, also with its characterization for 3D bioprinting, mostly of human tissues for regenerative medicine and disease modeling. So the idea was to combine all of these three technologies, the IPS cell technology that they had here in Kyoto, the development of non-viral vectors for gene delivery, and the production of 3D bioprinted airway epithelials that model the in vivo tissue of a cystic fibrosis patient. So yeah, that's pretty much it. We wanted to develop the non-viral vectors to transfect or to treat both the IPS per se, and also to use these IPS to generate a 3D bioprinted models. I don't know if this answers your question.
Yeah, no, okay. Absolutely. Yeah, and you also you also explained how these models contribute to the understanding of cystic fibrosis and the development of new therapies, especially since those are more biologically relevant. You already explained that. But regarding these new techniques, what new techniques, ways of working, or what have been your new approaches that you have found in this new laboratory that differ from the experience in Shanghai, like workflow, or perhaps like different methods or things that you can talk a little bit about us? How different it is working in Spain than it is working in Japan?
So I have to say that I haven't found very dramatic differences in the way of working in research, which is something that I'm glad to say, because that probably means that we're not doing it that bad. But or that we're all doing it the same. I don't know if it's good or not. But just I came here to learn about IPS and IPS technology and how to derive that to pulmonary lineage cells. So everything, understanding how these types of cells work, which is different from immortalized cells, how do they behave, how sensible they are to different stimulus, how can you characterize them, how all the workflows that come with the technology, and so just learning about that, and being able to in my head merge that with my knowledge, my previous knowledge from Spain, and our work, the work that we do there, how we can combine it, how can it be collaborative in some sort of way, and how can we make it better ultimately.
Perfect. And how would you compare in an international context from your perspective abroad? How would you describe the capabilities and the scientific level of Nanbiosis Unit 10? What is the current state of the international? What is the international context, and how does this Unit 10 fit into it?
So I think that when you're working in science, you have to rely on people that have different expertise than you do, and that is crucial. Good research centers have very good infrastructures and core services, and specialized technicians for each technique that allows you to rely those expertise in someone that knows more. For me, Nanbiosis, it's a platform that has that idea, and also expands it outside just one center, but in the whole Spain in this case. So I think it's crucial to enhance this platform, because it has a lot of potential, and put that in a very high power, because it's a very unique infrastructure, and I haven't seen that here. Here, those type of services are more in-house oriented, or in the research center, and each one has their own. So it doesn't allow that much of a communication within different research institutes, more than whoever I'm friends with, or whoever I'm already collaborating with. But you're not able to go for a technical issue to a platform that has a lot of researchers from a whole country, knowing very diverse techniques, and that can help you in different ways. So that is something that we have to promote and put a very high value on, I think.
Yeah, I fully agree. Well, that's the vision of Nanbiosis model, in fact. Regarding, well, you already mentioned that how Unit 10 specializes in non-viral vectors, and also how it focuses in advanced pharmaceutical formulations. And you already mentioned what can this state provide, what the state that you are carrying out, how it can contribute to your work back in Spain. How about the other way around? Why this type of expertise is particularly relevant when working with complex biological models? What can you provide in this lab that you find particularly useful in your personal expertise, or in the expertise of your lab back in Spain?
Yeah. So basically, IPS cells can be used for a lot of things, but one of the two most relevant are the in vivo regeneration of tissues by transplantation of the IPS, or partially derived to a tissue IPS. And the second case would be disease models. I think that in the first case, lipid non-viral vectors like lipid nanoparticles and other type of particles can act as a therapeutic strategy. You are going to transplant cells. You can genetically modify those cells to have a therapeutic potential or a therapeutic ability, and you can deliver that to the patient in a more safe context, because you cannot do this any other way. And in the case of disease modeling, this helps you to optimize these non-viral vectors prior to going to in vivo studies. So I really think it's two very complementary technologies that work very well, and that they can optimize each other very much. Goes both ways, I think.
Yeah, I think yeah, you're basically generating synergies between different technologies that can complement each other in a very nice way, actually, which highlights how important the international research stays like this one are for advancing science and collaboration. But let's talk a little bit about your personal level. How has been living and working in Shanghai? How has this
Kyoto
experience being in Japan, in Kyoto, influence the way you see your scientific career? Is there any change in your perspective, in your way of facing the research, academia, or your career in any way?
Yes. So right now I'm in my probably last year PhD, hopefully. And we're in a point where you have to start asking yourself questions of what do I do next? What do I like? What I don't like? What are my goals, my ambitions? And coming here and having the opportunity to know more things and know more people, know different ways of thinking, it's just it gives you a more wide view of the things that you can do, knowing that barely anything in this world is impossible. So just work for it and do your thing, and just to keep all your doors open, because you don't know when the next great opportunity is going to come from. It may come from Japan, it may come from Europe, it may come from Spain, but yeah, just, I don't know, having all your doors open.
I think you already answered my next question, but just in case, do you have any other extra advice for young researchers who are considering applying for international fellowships, or that are considering doing their PhD abroad, or doing research stays abroad, or what would it be for you? Any final thoughts that you may have to, if you're, let's say, what I always like to ask, if your younger self came to you going like, well, I don't know if I should go for a stay. Should I go? Should I stay? Should I
Yeah, stay or
stick to
should I
what I
go?
know? And why? What would you tell them?
I mean, I think that if you're a researcher, that means you're probably already a curious person. So just exploit that curiosity and see where it takes you. Always be persistent, talk to people. You will receive a lot of no's, of course, but in research and everything in life. So just be consistent with what you want. Don't be afraid to talk to people, to ask for help, to reach out, and I don't know, even with this call, like everyone, all the research that I knew that applied for the same grant that I did when they were postdocs, they told me, you're not going to get it. It's very competitive. Don't even bother. I was like, look, I may not get it, but it won't be because I haven't tried. So just go and try, and keep working on whatever that you're interested in.
Yeah, don't give up, guys.
Yes.
Well, thank you very much for your time, for spending some time with us
Thank
and
you.
contributing and telling us your story. It's always a pleasure, Lucia,
You
talking to
too,
you,
Ariel, of
and
course.
see you next time.
Yeah.